Welcome to the chapter Cell, its Structure and Functions.
This slide presents the overview of the chapter.
Now, let us study about the other important extra layer of the plant cell called the cell wall. Cell wall is the unique feature which is observed in plant cells. The main difference between the plant cell and the animal cell is the cell wall. The plant cell consists of a cell wall outside of the cell membrane, whereas animal cells do not contain cell wall. Characteristics of cell wall. It is tough and is the most important part of the cell. It is a flexible porous layer that provides a definite shape and cell protection. It exchanges substances continuously with other cells during its growth and development. It provides mechanical strength and support to the cell. It has narrow, tiny pores called pits and very fine strands of cytoplasm called plasmodesmata. Role of the cell wall in plant cells. The plant cells have greater resistivity over surrounding medium when compared to animal cells because the plant cells applies an inward wall pressure to resist the outward pressure exerted by the cell sap. Here, let us perform an activity to observe the nucleus in cheek cells. Click each tab to know more. To observe the nucleus in cheek cells. To perform this experiment, the materials required are a toothpick or ice cream spoon, permanent slide, cover slip, watch glass, needle, blotting paper, 1% methylene blue, glycerin, microscope, etc. Precautions. Do not scrape the cheek too hard because it may injure you. Scraped material should be spread uniformly on the slide. See that there should not be any air bubbles under the cover slip. Excess stains should be drained off on the permanent slide. Take a permanent slide and add few drops of methylene blue on it. Now take a toothpick in such a way that the flat end should be placed inside the mouth. Now put the toothpick inside the mouth and move the pick up and down for 12 times by sweeping against the cheek. Remove the toothpick from the mouth and place it on the die and spread the toothpick in the die to enable our cheek cells to spread out. Remove the toothpick from the die and cover it with a cover slip. Tap the needle on the cover slip to spread the cells. Place the permanent slide mount under the microscope. Observe the permanent slide under the low and high power of the microscope. Note the observations in your book. It is observed that the shapes of the cheek cells are irregular. It is observed that each cheek cell has a dark-colored border. It is observed that dark-colored spherical and oval-shaped structures are present near the center of the cell. The dark-colored outer edge of cheek cells is called cell membrane. The dark blue color inside the cell membrane is called cytoplasm. Dark-colored spherical and oval-shaped structures near the center of the cell is called nucleus. Nucleus is named by Robert Brown in the year 1831. Nucleus is the most important organelles of the cell. It is also termed as the cell's control room. Nucleus is the largest and is different from all the cell organelles. Schleiden, who proposed the cell theory, thought that new cells were created from the nucleus called cytoblast. Functions of nucleus. Nucleus regulates and controls all the functions of the cell. It determines the characteristics of an organism. It consists of all the genetic information. It is involved in the process of cell division. The nuclear membrane encloses the nucleus and separates it from its contents of cytoplasm. Nuclear membrane is similar to the cell membrane. Cells are categorized into types based on the presence or absence of the organized nucleus. They are eukaryotic cells, organized nucleus, and prokaryotic cells without organized nucleus. Eukaryotic cells. Eukaryotic cells contain a membrane-bounded nucleus. Almost all the eukaryotic cells have a nucleus. Red blood cells in some mammals and phloem sieve tubes in plants contain nuclei in the beginning and later thrown out of the cells and destroyed. Prokaryotic cells. Prokaryotic cells do not have a nuclear membrane to bind the nuclear material. Examples: bacteria, cyanobacteria, blue-green algae. Cytoplasm. In cells, the space between the plasma membrane and nucleus is called cytoplasm. It consists of various inorganic molecules like water, salts, proteins, nucleic acids, organic compounds, and different kinds of enzymes. Cytoplasm is made up of various cell organelles. These cell organelles are bounded by a plasma membrane. Each of these cell organelles perform a particular function for the cell. We have already learned that eukaryotes have membrane-bounded cell organelles, whereas prokaryotes do not contain membrane-bounded cell organelles. Functions of the cytoplasm. It helps in the exchange of materials between cell organelles. Breaking down of nucleus takes place in cytoplasm. It helps in intracellular distribution of molecules, enzymes, and nutrients within the cell. Biosynthesis of proteins, fatty acids, and nucleotides takes place in cytoplasm. Now, let us learn about protoplasm versus cytoplasm. Earlier, it was believed that the core of the life was stored in the fluid present inside the cell. Hence, it was named as protoplasm, which indicates life fluid. But after the inventions, it has become clear that fluid is just a medium in which different kinds of particles and membranes float around, and it is the function of the cell that actually takes place in these organelles. Thus, it is understood that life resides in these organelles. After the discovery of nucleus, it is understood that different materials so are present inside and the outside of the nucleus is different. Hence, protoplasm is renamed as cytoplasm, which is a cell fluid. The fluid present inside the nucleus is named as nuclear fluid or nucleoplasm. Now, let us learn about the important cell organelles in plant cell. Cell organelles perform an important function in the cell. There are many cell organelles that are found in cytoplasm of the cell. They are endoplasmic reticulum, Golgi apparatus, lysosome, mitochondria, ribosomes, plastids, vacuole. Click each tab to know more. Endoplasmic reticulum. When a cell is observed under an electron microscope, a network of membranes appear throughout the cytoplasm called endoplasmic reticulum, ER. This network creates passages in the cytoplasm to transport the substances from one part of the cell to other. The structure of ER membrane is similar to the plasma membrane. There are two types of endoplasmic reticulum: rough endoplasmic reticulum, smooth endoplasmic reticulum. Rough endoplasmic reticulum. Some granules-like structures are present on the surface of endoplasmic reticulum called ribosomes. Ribosomes play an important role in protein synthesis. This type of parts is called as the rough endoplasmic reticulum or ER. Proteins are manufactured in the rough endoplasmic reticulum. Smooth endoplasmic reticulum. If the parts or sections of endoplasmic reticulum do not contain ribosomes, then it is said to be smooth endoplasmic reticulum, SER. SER helps in the synthesis of lipids, fat molecules, which are important for cell function. These synthesized proteins and lipids are sent to various parts in the cell based on the need through endoplasmic reticulum. Some of these proteins and lipids help in building the cell membrane. Functions of endoplasmic reticulum. It serves as a channel and helps in transporting of materials between different regions of cytoplasm or between cytoplasm and nucleus. It acts as a cytoplasmic framework and provides surface area for various biogeochemical activities. In the liver cells of vertebrate, smooth endoplasmic reticulum helps in detoxifying many poisons and drugs. The membranes of endoplasmic reticulum are made up of number of enzymes for various metabolic activities and cytochromes that take place in electron transport. Golgi body or Golgi apparatus. In the year 1898, Camillo Golgi discovered this organelle by using a compound microscope. But its finer structure can be observed only under an electron microscope. Golgi apparatus is made up of several membrane-bound vesicles that are arranged parallel to each other in stacks called as cisterns. The proteins and other substances that are produced in the ribosomes reach the Golgi body through these vesicles. The distinct components that are visible in the Golgi complex are flattened sacs or cisternae, clusters of tubules and vesicles, large vesicles or vacuoles. The Golgi apparatus has convex forming phase and concave maturing phase. The convex forming phase receives vesicles from endoplasmic reticulum and the concave maturing phase produces secretory vesicles and lysosomes. The number of Golgi bodies varies from cell to cell. The number of Golgi apparatus are large in cells that secrete hormones and enzymes. Functions of Golgi apparatus. Golgi apparatus helps in secretion of mucus, enzymes and hormones. It helps in storage, packaging and modification of secretory products in vesicles. It helps in the manufacturing of complex sugars from simple sugars. It helps in the formation of lysosomes. Now, let us learn about lysosome. Lysosome. Lysosomes are membrane-bound sacs that are filled with digestive enzymes. These enzymes are made up of rough endoplasmic reticulum or ER. Lysosomes have a resistant covering membrane that protects the cell from digestive enzymes. Lysosomes help in the disposal of the wastage from the cell. Lysosomes maintain the cell clean by removing the worn-out materials and digesting the foreign or bacteria materials. Lysosomes contain destructive enzymes. If any bacteria enter the cell, lysosomes break them into small pieces. This is because the enzymes released by lysosomes are very powerful. Suppose, if the cell gets damaged, lysosomes burst and release an enzyme which digests their own cell. Hence, lysosomes are called suicidal bags of the cell. Functions of lysosomes. Lysosomes help in intracellular digestion. Lysosomes bring about cellular breakdown and are associated with aging. They provide energy during starvation. Mitochondria. Mitochondria is called as the powerhouse of the cell. Mitochondria is a membrane-bound organelle present in almost all the eukaryotic cells. These organelles act like a digestive system which takes in nutrients, breaks them down, and creates energy-rich molecules for the cell. The biochemical processes of the cell are known as cellular respiration. Now, let us learn about other important organelle, ribosomes. Ribosomes are small granule-like structures present in the cytoplasm of the cell. Ribosomes are present in both prokaryotes and eukaryotes except in mature sperms and RBCs. In eukaryotic cells, ribosomes occur freely in the cytoplasm as well as attached to the outer surface of the rough endoplasmic reticulum. In case of prokaryotic cells, ribosomes are found floating freely in cytoplasm. Functions of ribosomes. Ribosomes help in protein synthesis of the cell. Hence, they are termed as protein factories of the cell. Plastids. Plastids are cell organelles that are present only in plant cells. Plastids consist of various membrane layers that are present inside a material called stroma. From external view, plastids are similar to mitochondria. Plastids are of three types. They are chloroplasts, chromoplasts, leucoplasts. Chloroplasts. The primary function of chloroplasts is to trap energy from sunlight and transform it into chemical energy. This helps in carrying the process of photosynthesis. Each chloroplast is covered by a double-layered unit membrane. The inner membrane of chloroplast is made up of matrix or stroma and disc-like bodies called grana. Chromoplasts. Chromoplasts are colored plastids. Chromoplasts contain fat-soluble pigments. These are formed either from chloroplasts or leucoplasts. Chromoplasts are red, orange and yellow plastids present in fruits, petals and flowers of a plant. Leucoplasts. Leucoplasts are colorless plastids. These are named on the basis of substances they store. They store starch, oils and protein granules. Vacuole. Vacuoles are the storage bubbles found in the cells. It is a membrane-bound organelle found in both animal, bacterial, fungal and plant cells, but are much larger in plant cells. Vacuoles might store food or any variety of nutrients a cell might need to survive. They can even store waste products.
Knowledge check. Attempt the following questions to check your understanding. Knowledge check. Attempt the following questions to check your understanding. Knowledge check. Attempt the following questions to check your understanding. Knowledge check. Attempt the following questions to check your understanding. Knowledge check. Attempt the following questions to check your understanding. Knowledge check. Attempt the following questions to check your understanding. Knowledge check. Attempt the following questions to check your understanding. Knowledge check. Attempt the following questions to check your understanding. Knowledge check. Attempt the following questions to check your understanding.
From the image on the screen, identify the parts of the plant cell by dragging the labels onto the respective part of the plant. The first one is done for you. Click on the Submit button after completion to view the correct answers.
Select a suitable answer from Group B for Group A and drop it in the bracket provided against A. Read the questions and attempt the answers on your own. You can click Answer for your reference. Drawing skills. This section helps you to sketch images in a step-by-step manner. Click each tab to know more. Test your understanding of the chapter by taking the mock unit test